Okay, good morning. This week we are going to be doing lecture 4, which is membrane transport. So before we do the study guide, I would like to go through the idea of fluid mosaic model just as a review for you. So if you could please draw along, that would be great. Okay, so here is your cell. There's the cell, and then there's going to be a nucleus in the middle. And as you know, that is going to be the cell membrane. And the major component of the cell membrane is, of course, the phospholipid bilayer, right? And so we're going to zoom in to this location here, zoom in on a slice of the cell membrane, and we are going to draw it out, okay? So here is your phospholipid. There is the polar head, and then you're gonna have some hydrophobic tails. Okay, some of them might be saturated, some of them might be unsaturated. It's going to be a mixture of both. And depending on what kind of membrane we are looking at, sometimes you will have more unsaturated fatty acids. Sometimes you would have less. It really depends on the type of cells that we are looking at. Okay, but the key thing to know here is the membrane is composed of 2 layers of phospholipids. So that's why this whole thing is called a phospholipid bilayer. Okay. And remember, this is a single phospholipid molecule. The head, which is made up of the phosphate, that's going to be the hydrophilic part. Hydrophilic head, which will be facing the outside as well as the inside of the cell. All right, so in case you're a little bit confused with the orientation here, this would be the outside of the cell. Which we call the extracellular fluid, okay, ECF. Extracellular means outside of the cell. So really it's the fluid that's outside of the cell. Whereas over here we have the inside of the cell. Inside of the cell, which is the cytoplasm, right? So the hydrophilic head is either going to be facing the outside or the inside, both of which is going to be an aqueous environment, a water-based environment. And in the middle, these tails, that's going to be the hydrophobic tails. Okay, so by organizing in this way, it creates a thick barrier in the middle that prevents things from going through easily. Scattered throughout the membrane, there is going to be a bunch of proteins. Okay, so some proteins are going to go all the way through. It's going to extend from the outside all the way to the inside. Does anybody know what we call this kind of protein? What kind of membrane protein would this be if it goes all the way through? All right, that's okay. I will tell you. This is the integral protein. So maybe it forms a channel for things to go through. Maybe it's going to participate in some kind of signaling. Okay. Other proteins will just be attached either on the inside or on the outside. Okay. They don't really go all the way through. These one we call the peripheral protein. Okay. And so one goes all the way through, the other does not. Oh, I think I spelled this wrong. Peripheral protein. Okay. Any questions so far? What else is in the cell membrane? Does anybody remember what else is in the in this fluid mosaic model besides the proteins and the phospholipids? Yeah, Mavis says cholesterol. There are definitely some cholesterol. So we have some cholesterol here, maybe some cholesterol here. They will insert themselves in between the phospholipids. Okay, so these are cholesterol, which is a type of steroid, right? And the more cholesterol you have in the cell membrane, the more fluid it will be. So more cholesterol equals more fluid, right? More liquidy, right? And then less cholesterol equals to more solid. So depending on the type of cells we are looking at, sometimes you want a little bit more cholesterol to make the membrane more fluid, and then sometimes you want less cholesterol to make the membrane more solid. So here is a question to think about. Let me just type it out for you. Make it bigger. Okay, so consider a plant that grows in cold environment and a plant that grows in hot environment, like a desert or something. The cell membrane of the plant growing in the cold environment will have compared to the plant growing in the hot environment. Okay, so what is the answer? A, we'll say, oops, this is too small. Over here, we'll say A, more cholesterol, less cholesterol, or same amount of cholesterol. Okay, so what do you think the answer is going to be? Let me launch the poll. Okay, anyone else want to try? Okay. Here are the results. About half of you say A, half of you say B. So let's think about this. When a plant is growing in a cold environment, if you think about it, cold temperature makes things freeze, right, and become solid. If you put like butter in the or like oil, if you put oil in the fridge, they become more like a solid state, right? So similar situation is going to happen. If a plant is growing in a cold environment, their cell membrane is likely to become more solid. So you don't want the cell membrane to be too solid, right? Because otherwise things is not going to be able to go through, which is why in order to counteract that in the cold environment, you would want to put more cholesterol, right? To counteract that drop in temperature, right? So that it is not going to be too solid. And so if you look at the cell membrane of the plant growing in the cold environment, they will have more cholesterol compared to the plant in the hot environment. On the other hand, if you think about the plant growing in the hot environment, the higher temperature makes the membrane very fluid, very leaky, right? Because now the molecules are very energetic, they might be moving really quickly, so things can leak out, and you want to counteract that, right? To make it less fluid, you will need to decrease the amount of cholesterol. So plants in cold environment, definitely you would expect more cholesterol. Plants in hot environment, you would expect to have less cholesterol in the cell membrane. I hope that makes sense. There are other additional things, right? You might have sugar that are stuck on the protein, okay? And that makes it a glycoprotein, a sugary protein. Let me just choose a different color here. Glycoprotein. Right? You might have some sugar that are stuck on the lipid as well, and that will make it a glycolipid. Glycolipid. So these things are important when it comes to cell recognition, for example. Okay, so there you have it. That's the fluid mosaic model. Hopefully that's a good review for you. We're going to go back to our study guide now. And the first question asks us to describe the functions of membrane proteins. So there are several diagrams here. Let's go through them one by one. First one here, we can see this is your cell membrane. This is the phospholipid bilayer. This is the inside of the cell, and this is the outside. And so the first function for membrane proteins here is it allows the attachment to cytoskeleton. We learned about cytoskeleton last week. So all these green fibers that you see, these purple, orange fibers that you see, those are things that are like actin, microtubules, all those things. Well, some proteins has to be able to attach onto them, right, to form anchorage points, for example, and that would be the job of the membrane protein. So this provides attachment points and anchorage point. And by attaching to these proteins, we are able to, we can say anchorage point to extracellular matrix and intracellular matrix. That just means like all the fibers that you find outside the cell and inside the cell. And that will helps with cell movement. And maintains cell shape. Okay, that's the first function. The second function here, again, this is the inside and this is the outside, and these things, they are signaling molecules. Okay, such as a hormone. So this is going to be cell signaling. Okay. The membrane protein acts as a receptor for a signal molecule. Okay, so for example, it could be a protein hormone. Okay, steroid hormones, they can just go right through the cell membrane, right? But protein hormone, they cannot because it's hydrophobic in the middle. And then once the signal molecule binds the protein, the membrane protein, will trigger a series of events that change the cell's behavior. Okay. Now in the notes, in the PowerPoint slides, there's a picture, I believe. It looks something like this. There is the cell, and then we have the membrane receptor right here with the protein receptor on the surface. That's the receptor. And then you would have a signal molecule that binds to it, right? Let's say that's like insulin or something. Insulin will bind to it, and then that will trigger a series of events. Okay, so these series of events in the notes, they are called signal transduction. Okay, which would then, this is the nucleus, which would then cause something to change in the nucleus. Maybe you turn on a gene, maybe you turn off a gene. And in the case of insulin, you're going to put like special glucose receptors on the surface so that the cell can take in the glucose from the blood. Okay, but the key point here is the insulin never really got into the cell. They just bind to the receptor, and the receptor will cause a chain reaction that ultimately change how the cell behaves. All right. Next one. There's another picture. That is enzymatic activity. Okay, enzymatic activity. So enzymes that belongs to the same metabolic pathway are grouped together in the membrane. Not always, but sometimes. Yeah. So what does that even mean? Well, a metabolic pathway is basically a series of chemical reactions. So let's say, you know, we're going to take some reactant starting at A, and we need to convert it into B, and then B needs to be converted into C. In order for us to do that, though, we're going to need to have enzyme 1 here and enzyme 2 here. So rather than having enzyme 1 and 2 randomly located in the cell, well, let's put them right next to each other on the membrane. Okay, so when A comes, 1 is going to get converted into B, and immediately B can be worked on by number 2, which then gets converted into C, right? Okay, so this allows the reaction to be carried out rapidly. Okay, so this way, reactions can be carried out quickly near the cell membrane. There are several reactions related to immunity, for example, that needs to take place quickly near the surface of the cell. Okay, but that would be like one example of it. All right. Okay, next part right here, we have two cells. Again, not sure if it is obvious. So I'm going to label it for you. This is cell number 1. Cell number 1. This is cell number 2. Okay, and this is going to be one membrane protein, and this is another membrane protein. And you can see here, they are stuck to each other. So these proteins help you glue two adjacent cells together. Okay, so we call this, oops, wrong color. We call this intercellular junction. Okay. So two adjacent cells connects two adjacent cells together, which prevents things from crossing between cells. Right? So if you imagine these cells are like part of your small intestine or something, you need to close the gap between them so the food's not going to leak between them, right? Okay. So it prevents uncontrolled movement of material across the cell. Okay, next one here, we have cell-cell recognition. Okay. So there are many antigens on the surface of a cell. Okay, what are antigens? Antigens could be proteins, right? Glycoproteins, could be lipids, right? So all these different molecules create a pattern that is on every single cell. And the antigens you have will be different from the antigens of someone else, right? No two people in this world is going to have the same set of antigens, right? Some people will have more similar antigens than others if they're related. Okay? If you have a twin, identical twin, then you would have the same set of antigens on the surface. But generally speaking, unless you have an identical twin, no one will have the same set of antigens as you. Arthur asked what is antigen. So antigens are proteins, sugary proteins, lipids that are on the surface of a cell. So you have a bunch of molecules that are on the membrane. Depending on what kind of molecules you have, that would determine what your antigens are. Okay. For example, right, the ones that people are most familiar with would be, if you think about your red blood cell, if you have protein A on the surface, then your blood type is type A. If you have protein B on the surface, then your blood type is type B. If you have both protein A and B on the surface, then you're type AB. That's just one example, right? Okay, so everybody's going to have different antigens. And so, when a bacteria goes into your body, the antigens that are on the bacteria will not be the same as what you have inside your cell, and your immune system is going to immediately recognize those foreign antigen and start attacking them. Okay. Same thing with like organ transplant, right? You cannot just take anybody's kidney and then give it to someone else. The body is going to reject it because the antigens are different, and your immune system see these different antigens as dangerous, and then they're gonna, basically try to get rid of them. So the membrane protein is going to be part of those antigens. Does that make sense? Any other questions? All right. And finally, last but not least, we have this final function, which is transport. Transport. Okay, so membrane proteins will participate, will help in movement of substance across the cell membrane. Okay. For example, facilitated diffusion. Okay. So those are the functions of the membrane proteins. Okay, let's see if we have some poll questions. Not yet. Okay, we'll come back to this. All right, next page. Complete the following diagram to illustrate the relationship between anabolism and catabolism. So just quick summary here, quick definition for anabolism and catabolism. Anabolism is basically building things, building big molecules, building macromolecules. Okay. Whereas catabolism is breaking down macromolecules. Okay. So over here, we have a bunch of macromolecules, and they are macromolecules that are found in our food, for example. Okay. So macromolecule found in food. If you eat pasta or rice or bread, then there's going to be carbohydrates in them. We learn about starch being a macromolecules, right? There will be lipids in it. There will be proteins in it. All those are macromolecules in the food. So once we eat them, it's going to go into our body, and we will digest them. And eventually, we're going to have to break down the food molecules on the molecular level. And to do that, we are going to be doing what we call catabolism. Okay, so over here, catabolism. Okay, or catabolic, B-O-L-I-C, catabolic reactions. Okay. And in doing so, we are going to be able to extract the energy from the food molecule, and we will capture those energy in the form of ATP. Okay, so ATP, for those of you who don't know, ATP is the usable form of energy. Usable form of energy in cell. And it stands for adenosine triphosphate, right? But the capture of the energy is not very efficient. So a lot of the energy that is being released by these food molecules is actually wasted as heat. Right? So in the process of doing this, you're going to release a lot of heat, which is okay because we need to maintain a body temperature of 37 degrees Celsius, right? So the heat is going to keep us warm. Right? If you think about a light bulb, right? Not the new kind are more efficient, but like the older ones, that looks like this, right? And if you leave it on for a little bit, you touch it, it's actually going to be hot, right? And that is because the light bulb is inefficient, right? The job of the light bulb is to convert electricity into light, but a lot of that is dissipated as heat. So our body is the same. The goal is to convert the food into usable form of energy, ATP, but we're not very efficient. We are about like, 30, 36, 37% efficient. And much of what we try to capture is lost as heat. So in a way, we kind of like the light bulb. And then we're going to end up having all these building blocks, right? So these building blocks could be amino acids that you get from breaking down the proteins. It could be like monosaccharides, right? It could be fatty acids, right? A bunch of building blocks, which we then could could use them to build molecules that we need. And to build molecules that we need, we're going to use the anabolic reactions, right? Or anabolism. And finally, we will get molecules that are needed in the body. Molecules needed in the body. Okay. So again, example, we might need to have some insulin, right? We might need to have some testosterone or something. So your body will take these things and build them. Right? So there is the relationship between catabolism and anabolism. In essence, what you should know is that the energy released by catabolism is used to drive anabolism. Okay, so to build things, we need energy. Where do we get the energy from? From breaking down the food, right? So one drives the other. Of course, we use ATP for other purposes as well, but this is one example of how we use it. So collectively, all these reactions, the catabolic reactions, the anabolic reactions, all these things are going on within the body. We call that metabolism in general. Okay. Anybody have any questions? Let's do a poll. See if you were paying attention. Okay, go ahead. Oh, sorry. This is supposed to be select all the correct answers. I launched the wrong poll. My bad. Let's try that one more time, please. All right, let's see. Catabolic reaction. So that is the one where we are going to be breaking things down. Consumes more energy than they are produced. That is not true, right? The whole purpose of catabolic reaction is to allow us to create ATP, right? So if the reaction takes up more energy than they produce, then we wouldn't be left with any ATP at the end, right? So it should be the other way around. Okay? They actually produce more energy than they consume. Breaks down polymers into monomers. That's correct, right? We are breaking down the macromolecules. Produce excess heat to warm up the body. Yes, it was written down in our diagram. Only occurs in human cells and not other cells. That's not correct. You know, all living things have these kind of reactions. And so the answers are B and C. All right, so let's go back here and continue. List 3 different kinds of cellular work that requires ATP. So let's quickly you know, go through the concept of ATP again. So ATP is adenosine triphosphate, right? So adenosine is like as in like the adenine in DNA, right? And so we'll call this the adenine, and it has 3 phosphate in it, P, P, P. And the bond that connects the last 2 phosphate is a high energy bond. Okay, that's a high energy bond. And what happens is when you break off that P, that last phosphate, that high energy bond is going to release a tremendous amount of energy. Okay, so that energy is going to come out, energy, and we can use that energy to do various things. Okay, and that's what this question is asking. What are we using the ATP for? Okay, so the energy can be used to do different work. We'll come back to this in a second, but after you break up the P, you will be left with just the A and then 2 P's, right? And then just 1 P floating around. So this is ATP, adenosine triphosphate, but afterwards we are going to have ADP, adenosine diphosphate, plus just a phosphate. Okay. So how do we add the phosphate back onto ADP? Well, something has to supply the energy to recharge the molecule, right? So in order to recharge the molecule, we're gonna need to have catabolic reactions. So by breaking down our food, that energy is then captured by ADP. We're gonna put the phosphate back on, and now it's ready for another round. So it just keeps on cycling between ATP and ADP, and you'll be able to use the energy for various needs. Okay. So what are we using it for? Can anybody tell me what are some examples of cellular work that requires ATP? Patricia said mechanical work. That's correct. Okay, so what exactly does mechanical work means? Mechanical works means there is some kind of movement involved. Movement. So maybe the cell needs to move, physically moving your arm, that requires ATP. Okay, what's another example? Michelle said facilitated diffusion. Yep, so that is transport work. Transport work, although facilitated diffusion does not actually need ATP, but there are some kind of transport work that does require ATP. We call them active transport. We will explain more about facilitated diffusion later on. And finally, Mavis says chemical work. Okay, so for example, anabolism, right? Okay, so these are all the different parts, different things in the body, in the cell, that requires ATP. Okay. So, if you imagine yourself running at full speed, as fast as you can, you won't be able to maintain that speed forever, right? Like, you probably can do that for a minute or 2 minutes, but eventually you are not going to be able to keep up. And one of the reasons why you can't do that anymore is because the demand for ATP is too great, and your body don't have enough. You're not able to, replenish the ATP quick enough, right? And you build up a pool of ATP, and that causes your cells to not be able to do mechanical work anymore, and you have to stop and recharge, so to speak. Okay, that's that. Moving on, list 4 factors that affects the rate of diffusion. So let's again define what diffusion is again. Anybody want to tell me what diffusion is? Probably easier if you unmute and tell me than typing it up. But I'm sure some of you knows what diffusion means. What is that? What does that mean? Okay, Sandeep says size. Yeah, that's the answer. But can anyone tell me what diffusion means? Passive flow across membrane. Yeah, go ahead, please. See someone, Michelle, you unmute. You want to explain?
Diffusion is going substance from high concentration to low concentration.
Yeah, exactly right. Thank you. So that is the movement of substance from high to low concentration. Okay, and this is a spontaneous process. It does not require energy. So how fast can the diffusion occur? Well, it depends on various factors, right? So Patricia say, Sandeep say, size. That's right. So size will definitely affect how quickly this occurs. Generally speaking, smaller equals faster, which kind of makes sense. If it's small, it's easier to move quicker, right? What's another factor that affects the rate of diffusion? Okay, we have a couple people say temperature. So generally, higher temperature equals faster, unless never mind. No one necessarily. I don't want to confuse you. Okay, so higher temperature means faster because the molecules are moving faster. They're more energetic. Another one is steepness. Okay, so the steepness of concentration gradient, right? Concentration gradient. Well, what does that mean? Well, if you think about physically a ball rolling down the ramp. Okay, so there is the ball. There's the ramp. Okay, and then there's another ramp. Okay, and then I put the ball. I put the ball at the very top. Okay, this is ball A. This is ball B. Which one is gonna roll down faster? Is it ball A or is it ball B? Which one will go down the ramp faster? Well, hopefully, you will say it is ball B, right? Ball B is going to go down faster, right? Because the difference in height from here to here is greater than the difference in height here, right? In other words, the difference here is steeper than over here. So that's the same thing when it comes to concentration gradient. For the purpose of explaining, let's say I have two chambers, okay, and this one is 20% concentrated versus here is 70% concentrated. So the difference, the triangle means difference, the difference is going to be 50%. Whereas if I have two chambers here, and this is 60%, 70%, the difference here is only 10%, right? So diffusion is going to happen faster when the difference between the concentration of 2 points is greater compared to in this case, okay? If the difference is very subtle, then there will still be some diffusion, but it wouldn't be happening too quickly, okay? So that's what it means by concentration gradient, okay? And there is one last one, I think Mavis mentioned it. It is the charge, okay? So, you know, things could be positively charged, things could be negatively charged. And the general rule is like charges repel each other, right? So if you have a bunch of positives, they're going to repel each other. So let's say I have a chamber here, and all this is important, you know, next class when we learn about metabolism. By the way, that's going to be my last new lecture with you, right? I think we're also going to have a test review. Or are we? Let me double check and come back to you. So here is the membrane, okay? And let's just say you have a bunch of positive charges here, okay? So what happens is, because there's high concentration here and low concentration there, they will go through. But in addition to that, all the positive charges will be repelling each other. So they would have an additional push, right? To make these positive go to the other side. Okay, so charge will definitely play a role in terms of the rate of diffusion. Okay, so here's a question that you might see on the test, and it's worth thinking about because people usually get it wrong. This is the same diagram that you see in the PowerPoint. So there's a U-shaped tube, and we're going to put some water in it. Okay, so this is going to be filled up with water like that. Okay, we're going to put a membrane here at the bottom. And this membrane only allows water to pass. Okay, nothing else. And let's say we have these solute that are dissolved in it, right? Okay, these red balls here, they are solute. Okay, so these are solute is what's being dissolved in the liquid, right? Solute. Okay. So what will happen over time? Okay, what will happen over time? Is it A, water level on the left increase? Okay, or is it B, water level on the right increase? No change in water level. All right, let me launch a poll for you. Okay, you think about that. Okay, anyone else? Just gonna give you a couple more moments. Okay, so let's see what's the correct answer for this. So, the idea here is that the red dots are not able to move because this only allows the water to pass through, right? So the water will move towards the side that it has higher solute concentration in order to balance it out, right? So one of the way to visualize this is if you put a spoon of salt in your mouth, then the salt is going to suck out all your saliva, right? So the high concentration of the salt is going to draw water towards that location. So because we have more solute here, the water will get pulled to the left side. And over time, what's going to happen is, the water level will rise here on the left because they're coming over. And then the water level on the right is going to drop. Okay. Does that make sense? For people who chose B and C, do you understand my explanation?
Yeah.
Okay, thank you. And if the rest of you needs more clarification, please do let me know. All right. So let me take a moment here and talk about the quiz next week. Okay, so the quiz is going to be on Tuesday, June the 5th. And again, availability will be from 6 to midnight, essentially, 45 minutes, and it covers lecture 3 and 4. So what we did last week and what we're doing right now. And here is the breakdown of the question types. Okay, so there are some multiple choice, some multi-select questions with more than one correct answers, some application questions. Okay, and that will be worth 10% of your final mark. and it's worth a total of 30. Okay. And also, I was just checking the course calendar. So next week we are going to have the 5th lesson, and then after that you will have the test. Yeah. So we'll do a review on the following week, and then that would be my last week. Okay. So no changes next week. Next week I'll still see you on Thursday, and then I'll remind you what's going on in week 6 later on. What are application questions? Application questions are also multiple choice, but they are like you know, like the like something like this would be like an example of an application question, because it's not straightforward, right? Like you actually have to apply what you have learned, right? Something like that cholesterol question that I asked at the beginning with the plant and the cold and hot environment, that's an application question. So it's something that's like a little bit harder, a little bit different. You have to apply your knowledge. That's what they are. Okay. All right. Describe what happens to the cell in each of the following solutions. On E Centennial, it says the quiz will be on the 7th. Oh yeah, it is the 7th. It is the 7th. What did I say here? This is 7th, not the 5th. 7th is the Tuesday, right? I just got the dates wrong. Yeah, thank you for correcting me. I'll post this on eCentennial afterwards. Okay, so it will be correct when I do that. All right, over here, we have 3 types of solutions. First one is isotonic. Okay, isotonic. What is the meaning of isotonic? Isotonic means the concentration of the solute inside the cell and the concentration that is in the surrounding would be equal. Okay, so that's the first thing to know. Let me just write that down for you. The concentration, square bracket means concentration. Okay, so concentration of solute inside equals to concentration of solute outside. Okay. And because there's no difference in concentration, the rate at which the water enters the cell will equals to the rate at which water exits the cell. Okay. So rate in, the water coming in, equals to the rate of water going out. So as a result, they cancel each other. Then we have no net water movement. Okay. In other words, if there's no net water movement, if you don't have more coming in or more leaving, then the end result is no change. No change in cell size. Okay. If you are an animal cell, this is the best. Okay, they're happy in the isotonic environment. If it is a plant cell, okay, they're okay. They are not very happy, but they're okay. They're not like, you know, sad or anything. they will survive in the isotonic environment, but this is not the best environment for them. Okay, so to like visualize this, you can think of, you know, having a bucket, and then here's your bucket, and you're gonna have a tap here. Okay, so there's already some water in the bucket, and then you turn on the tap, the water is gonna come in. So in this case, what's gonna happen is we're gonna have a leak in the bucket. We're gonna make a hole in the bucket. But what happens is the rate at which the water is leaking is exactly the same as the rate at which the water is coming in. So the water is coming in, and then the water is leaking here. Okay? But if they're the same rate, then what's going to happen to your water level? Right? It's going to be no change, right? So that's basically what happens here. The rate at which water comes in is exactly the same as the rate that it's leaving. So no change in the cell shape. Okay. Next one is hypotonic, HYPO. In hypotonic, you have a lot more solute inside than outside. Okay? And as we explained with the U-shaped tube, when this happens, you will have the water moving towards the side that has a higher solute concentration, right? So you end up having a lot of water coming in and it's leaving. Okay. So what's the situation here? Well, the situation is that the concentration of, too many colors, I'm confusing myself. The concentration of solute inside is greater than, it's higher than the concentration of solute outside. Okay, and as a consequence of this, the rate in is greater than the rate of water leaving. And so if you think about our situation here, it's like turning on your tap much bigger, like that, than it's leaving. So the water level will rise, right? Okay, and eventually it will overflow. Right? So the water will come in and the cell will swell and eventually it might burst. So cell swells and might burst. Right? We'll say cell swells. Okay, cell swells up, and then what happens? Well, if it's an animal cell, this is not good. Okay, they are going to burst or lyse. Okay, they will blow up and die because it's too big. It's like overflowing. Blowing into the balloon too much air, it pops. For the plant cell, they actually love this. Okay, they love this because the cell wall protects them. No burst, no bursting. Okay, and that makes the plant really hydrated, and then the plant is gonna prop up. You know, if you have any house plant and you forget to water them for like a couple of days, you can see the leaves like all withered, right? Okay, but as soon as you put water in them, the leaves open up again, right? So the ability to open up like that, to stand up, that's because of the hypotonic solution, right? All that extra water is going to swell up the plant cell, fill up the vacuole, and the plant is doing the best when they're in a hypotonic environment. Okay. Lastly, we have the hypertonic environment. Okay. Hypertonic is the opposite, right? You have more solute outside than inside. So in this case, the water coming in is not going to be as much as water leaving. Okay, so you have a lot of water leaving like that. So in our analogy here, that would be like, you know, really turning down the tap. Okay, really turning down the tap, but then you make a bigger hole at the bottom. So you're leaking way more than you're bringing in. And so the water level will drop, right? So over here we have concentration of solute inside is less than concentration of a solute outside. And so the rate coming in is less than rate out. Okay, so in both cases here, for plant and animal, the cell will shrinks. Okay, and we call this plasmolysis. Okay. So the plant cells not going to be happy. Animal cells not too happy. Okay, either. They're not like sad, not like bursting up sad, they're like, not too happy. We could do better. Okay, but plant cells, definitely not happy. Okay, if you leave them in hypertonic solution for too long, they would die. Try sprinkling salt onto into your soil. The plant will die, right? Because of this. Okay, any question about all these things? So to distinguish hyper and hypo, just remember hypotonic has an O in it, right? Big O, hypotonic. So this is like all the water coming in to filling up the cell and bursting. Whereas hypertonic, right? There's no O here. Just visually, it might help you remember it. Any questions? Okay, let's take a quick break and we'll do more in 5 minutes. Okay, see you in 5 minutes. Hello, let's keep going. Next question, we have a chart that compares active transport and passive transport. So for active transport, it involves the movement of molecules from low concentration to an area of high concentration. Okay, from low to high. And that is against concentration gradient. Okay. Whereas for passive transport, it is going to be going from high concentration to low concentration. Okay, which is going to be going down the concentration gradient. For active transport, yes, you do require ATP. And for passive transport, no ATP is required. Examples of active transport include protein pump. Right? So the membrane protein will actively pump the molecules across the membrane. And another example would be bulk transport. Phagocytosis, pinocytosis, that sort of stuff. And for passive transport, example would be simple diffusion. Okay, or facilitated diffusion. Okay, that is that. Anybody have any question? Okay, let's move on to the next page then. Give me 1 second. Something just popped up on the computer. I need to get rid of that. Why don't you do a poll here? And we'll see. Go ahead. Yep, I think this is pretty straightforward. I hope it's pretty straightforward. It will be movement of substance down its concentration gradient. Very good. Okay, let's see if we have another one. Go ahead. All right, let's see. When a cell is placed in a hypertonic solution, oh, sorry, I launched the wrong poll again, so I keep on doing that. I'm supposed to check all correct answers, but that's okay. Let's take it up. Okay, so hypertonic solution is when it shrinks up like that and all the water leaves. So net movement of water out of the cell. That's correct. The cell will not burst because the water is leaving, so the cell will shrink. And it will not burst, so this is not correct. Solution concentration is higher on the outside compared to the inside. That is also correct. That's the reason why the water is leaving in the first place, right? Okay, so the answer is A, C, and E. Again, sorry for launching the wrong poll. Okay, let's keep going. So here, choose the more concentrated solution in each of the following. The first one, we have 80% water and 20% starch versus 90% water and 10% starch. So obviously the one, you're looking for the one that's more concentrated, okay? So obviously the one that has more starch is going to be the more concentrated one. So over here, next one, 5 grams of salt in 50 grams of water versus 5 grams of salt in 25 grams of water. It's hard to compare when the amount of water is different. So what we can do is we can change it, right? Let's take this one, 5 grams of salt in 25 grams of water. And we want to make it the same as the other, right? So how do I make 25 grams into 50 grams? Well, I can just times 2, right? So that will become 50 grams of water. But if I multiply the denominator by 2, I have to multiply the numerator by 2 as well. And that's going to give me 10 grams of NaCl. Okay. So 10 grams of salt in 50 grams of water compared to 5 grams of NaCl in 50 grams of water. This is going to be the more concentrated one. Last one, 85% solvent, 15% solute versus 75 solvent and 25 solute. And of course, the one that has more solute is going to be the more concentrated one. Okay, so there you have it. That's how you do it. List 3 types of bulk transport. So anyone have any suggestions? 3 types of bulk transport. What are some answers? And that is to bring in large amount of substances or to expel them from the cell. So what are some examples of bulk transport? Someone on the iPhone says exocytosis. Good. Kathleen, Ryan, phagocytosis. Yeah. Okay, and then this is a type of endocytosis. What's another type of endocytosis besides phagocytosis? This is to bring in solid, right? This is cell eating, and we also have cell drinking. Anybody know what that one's called? Cell drinking is pinocytosis. Pinocytosis. All right. Any requests for these long answer questions? Okay, number one, describe how the sodium potassium pump works. Well, the main thing you should know about the sodium potassium pump is that on the cell membrane, you would have the pump. This is the sodium potassium pump. And it basically uses ATP to pump sodium out. So for every 3 sodium that you pump out, you will be able to bring in 2 potassium. Okay. So the key idea here is if you keep on doing this, you're going to end up having a lot of sodium on the outside, right? So you would have high sodium concentration on the outside and low sodium concentration on the inside. Whereas the potassium would be the other way around. When you keep on bringing the potassium in, you will have high concentration of potassium on the inside and low concentration of potassium on the outside. So that's why this is an example of active transport, because you have to keep on pumping the sodium out against its concentration gradient. And similarly, you have to keep on bringing the potassium in from low to high concentration. So this whole process of exchanging the sodium with the potassium, that requires ATP. Okay? And that's pretty much what you have to know. This is an active transport. Does that answer your question? So Karina, which particular one do you want me to go through? C? Okay. So endocytosis and exocytosis, how are they similar? Okay, so they are both examples of bulk transport. Okay, they both involve bringing in a large amount of substances or expelling a large amount of substance. They rather than moving individual molecules, you are moving them in large quantity. Okay, so both examples of bulk transport, both require ATP. Okay, and also both involves the involvement of the cell membrane. Okay, so let me try to show this to you. You can just watch. You don't have to copy. So there is a cell membrane. And then let's say there is some stuff that you want to bring in, right? So what happens is the cell membrane will surround those materials. Okay, these are series of diagrams. And what happens is afterwards, it will close up. And then that's how you bring it in. Okay, so the cell membrane will move, the cell will move towards whatever you want to bring in, and then it will cave around the substance and then form a bubble, and you will bring it in. So this is a vesicle. Does anybody know where this vesicle is going to go? Which organ now is going to receive this vesicle? Yep. Carl said Golgi. It could go to Golgi apparatus where it gets processed, or it could go and fuse with the lysozyme, right? Where it's going to be a lysosome, where it is going to get processed. Okay. So that's the similarity between endocytosis and exocytosis. Exocytosis would just be the other way around. Okay, like if this was a vesicle that's fresh off from the Golgi and you want to release the content, then it's going to go the other way around. Okay, and how are they different? Endo is to bring things in. Exo is to bring them out. Okay. Any other ones besides C? Okay. Oh, E, sure. Phagocytosis and pinocytosis. Again, how are they different? They are both examples of endocytosis because you're bringing things in. Okay, both are endocytosis. Okay, and therefore they're both bulk transport. They both require ATP. Okay, but phagocytosis is to bring in solid particle. Okay, like you eat up a bacteria or something. That would be a phagocytosis. Pinocytosis is primarily bringing in fluid. So there's not much substance to bring in. Right? You're bringing in all these cellular fluid from the outside, the extracellular fluid. So that's why this is cell eating. That's what it means. And this means cell drinking. Cell drinking, cell eating. Does that answer your question? Perfect. Okay, let's try these application questions. In 2007, a woman who competed in a radio station contest to see how much water she could drink without going to the bathroom died of water intoxication. This was an actual thing that happened. You can look it up. Explain why drinking a large amount of water in a short period of time could result in water intoxication. Okay, so anybody want to give me some ideas of why drinking so much water is going to be toxic for the body? Red cells become hypotonic. Yep. So you're gonna create an hypotonic environment, right, in the blood. And what's the concentration? What's the consequences of that? Well, you will have your RBC, red blood cells, will burst. Okay, so if you have a lot of red cells bursting, then you will have, for example, anemia. Okay, cannot transport water, right? Oxygen. Cannot transport oxygen, and you could die from that. What's another reason? High concentration of calcium. Why? Why would drinking a lot of water increase the concentration of calcium? Oh, not quite. Water actually doesn't have, calcium in it. Water doesn't actually have much of anything in it. Unless you're drinking mineral water, then that's a different story. But if you're just drinking regular water, then very small amount of electrolytes in it. But good try though. In fact, what drinking a lot of water will do to you is it's going to dilute the electrolyte in the body. Okay, so electrolytes would be like sodium, chlorine, potassium. All these things have different functions in the body, right? Calcium. So we need the sodium and we need the potassium for communication of nerves. We need the chlorines for blocking certain communications in the body. We need calcium for muscle contraction. So if you're diluting all these electrolytes, you will lose all those functions. and that could be a problem. Okay, so drinking huge amount of water in a short period of time is not a good idea. Number 2, single-cell organisms that lives in fresh water are constantly exposed to a hypotonic solution. Speculate what kind of transport mechanism could be used to prevent lysis. Okay, so we talked about this thing called the paramecium, right? Last time, it's a single-cell organism that lives in a pond, and water is going to keep on coming in, right? If it doesn't have some kind of a mechanism to counteract that, it's going to burst. So the paramecium actually has a special pump, okay, a special vacuole that will take all those water and it will pump it out. Okay, so use active transport to pump out excess water. Okay, so that would be an example of how it uses ATP to counteract the water coming in. Salting has long been used as a method of preserving food. Coating meat with layers of salt will dehydrate the meat as well as any microorganism. Explain how this process works. It's pretty self-explanatory. If you put a lot of salt, it sucks out all the water from the cell. And if you have any microorganism that is on the meat, they will get dehydrated and die. And when you don't have any microorganism, then the food will not go bad. And so before refrigeration was invented, salting was the primary way of preserving food, over long periods of time. But eating a lot of preserved food nowadays is not healthy, right? We don't use salting to preserve food anymore. We use preservatives, things like sodium benzoate. We use things that contain nitrate to preserve food, right? So all those sausages, ham, all those stuff have high concentration of nitrates, and nitrates, if you consume it regularly, it has been tied to an increased risk of getting various forms of cancer, colon cancer, for example. So it's not very healthy. All right. Anyone have any questions before I move on to the fill in the blanks? Sandeep, you have some questions? I see that you unmute. No? Okay. All right. Here we go. The plasma membrane contains 2 layers of, the answer is phospholipids. Cholesterol are important for maintaining the proper fluidity of the plasma membrane. Metabolic reactions that break down molecules into smallest unit, that would be the catabolic reactions or catabolism. Whereas anabolism is the other way around, where you will build molecules from smaller units. And ATP molecules contains adenine, some kind of sugar molecule, and 3-phosphate. The diffusion of water is called osmosis. Carrier protein and channel proteins are examples of facilitated diffusion. They just provide a way for things to go through. An animal cell will shrink when placed in a hypertonic solution. A hypotonic solution has lower solute concentration than the inside of a cell. Cell eating is called phagocytosis, whereas cell drinking is pinocytosis. Finally, if you place the cell in a hypertonic solution, it will undergo plasmolysis. Okay, so those are the answers for the fill in the blanks. I have a couple more poll questions. Let's go through them. Go ahead. Okay, so the easiest one to cross would be something that's small. So anytime you see large, we can just, ignore them. And if it's hydrophilic, it's not going to be able to cross easily. Hydrophilic or charged particles, they will not cross the membrane easily because remember the center of the membrane is hydrophobic, right? So you have a thick layer of hydrophobic barrier in between. So anything that has a charge or it's hydrophilic will not be able to cross. To bring in a bacterium, that requires bulk transport. That's phagocytosis. It's not going to be able to cross easily. So the one that will cross the easiest will be something that's small and something that's hydrophobic. Okay. This one is really long, but it's an example of an application question. I will just go through it with you. Okay, so a student is designing an experiment to show the effects of the movement of water across a selectively permeable membrane. He placed a highly concentrated salt water solution into a bag that is permeable only to water. So let's draw this out and see what happens. Okay, there is going to be a bag right here, and it's highly concentrated salt water, okay, compared to the outside. Okay, so outside is pure water, so you actually won't have any of these dots. Okay, pure water. The question says, right? Which of the following can be expected? Well, could you please zoom in on the question like that? That's as far as I can go, sorry. Let me just put this here and zoom in, make it smaller. Yeah. Are you doing this on the phone by any chance? I guess it would be a little bit smaller if it's on the phone. In any case, let's think about what happened. Well, over here, because it's hypotonic, you have a higher concentration on the inside. There's going to be a lot of water coming in, right? Okay, so the volume of the bag will decrease over time. That's not true because of all the water coming in. The pH of the content of the bag will increase over time. Well, this has nothing to do with pH, right? pH only changes if you have hydrogen moving, right? Salt will leak from the bag until the concentration inside the bag equals the concentration in the beaker. That's not true because it's only permeable to water. The net flow of water will be from the bag to the surrounding of the water. That's not true either, right? Because you have all the salt inside, the water is supposed to come in. So the net flow of water will be from the beaker into the bag, causing the content of the bag to increase in volume. That is the correct answer. Okay. All right, last question. I will let you do this one. This one is quite short to read, so you can do it yourself. Go ahead. Okay, thank you for answering the question. This is a trick question. It's meant to, you know, trick people, I guess. But hopefully after I explain it, you will understand what the correct answers is supposed to be, and you will get it right next time you see this. So after freezing and thawing some animal cells, what would happen if you place it in a hypotonic solution? I understand why many of you chose this one as the answer, because you're like, oh, hypotonic, meaning water will come in, it will swell, and it will burst, right? But it's an application question. That means you have to think a little bit more. So the freezing and thawing is actually going to be important in finding the right answer. What happens if you freeze liquid? Right? If you fill water up in a plastic bottle, you put it in the freezer. What happens the next day to the water? How does it look compared to before? Well, yes, it's solid, yeah, because it's frozen. But like anything else that you can tell me about? Like, how does the water bottle look? Sure, we all had this experience, right? Like, you know, it's super hot outside. You want to freeze some water, just put it in a plastic bottle. The bottle looks like it's gonna explode. That's right. Okay. In other words, the water expands, right? Okay. So if you think about a cell, and you freeze it, right? It's going to expand and it's going to burst. The cell is going to be full of holes, right? And you know this because I don't know, maybe you don't know this, but if you ever freeze vegetables, or if you buy like frozen vegetables, they tend to leak out a lot of the green stuff, right, from it. That's because, again, the freezing, the ice crystal actually will puncture the cell wall, and it will, you know, make holes all over the cell membrane. So after you thaw it, it's already full of holes. And if you put it in a solution, it actually doesn't matter if you put it in a hypertonic, hypotonic, or isotonic, because it's full of holes. You know, the water is just going to go in and out, right? And the solute, right, is going to go in and out. There's nothing that's regulating anything. So as a result, the animal cell will not change size. Does that make sense? Right? Essentially, the freezing and the thawing destroy the cell membrane. Okay, and there's no longer any kind of regulation, and so it's not going to swell up or shrink. Is it okay for all of you who chose like A? I hope this makes sense, and you will choose the right one on the test if you see this question. Okay, well, thank you for coming. We will do lecture 5, which is the last lecture of unit 1. Okay, next Thursday. It's a very difficult lecture, especially if you have not learned it before. So please watch the videos, watch the recordings before you come to class. Okay? I know not all of you have watched it, but it's important, especially important, that you do that before coming to the next tutorial. All right. Okay, so enjoy the rest of your week. I'll see you next week. Bye for now.